Now let's go back to this checklist part here, metadata checklist, and let's download the checklist together. We will need this file to run a FAIRe, the template generator, which I will demonstrate very shortly. So you can click this download tab here or directly just to this download page here. In this page, you can find the latest version of checklist as well as other resources such as template, the full template, codes and tools, and example data sets. Now we're gonna go into here and then it will take you to this GitHub page and you can download using this link here. Let's open this checklist that you just downloaded now into your computer. You will find the two tabs here, README and Checklist. In the README tab, you can find some of the instructions of data formattings and then the terms and definitions here. So some of the terms that we used in our checklist that might be a bit confusing, so we define this here. Now let's move on to this checklist tab, and I'm going to expand some of the columns so that you can see it better. I'm going to quickly go through this checklist table, the structures, so you can understand how to follow this. There are column C is the term names, so this is basically the term names or column headers of your metadata or different types of data, which are also described here. So these terms should go into project metadata, while sample name can go into multiple data types such as sample metadata, amplification data, standard data. Those are for targeted assay data types. And then if you are doing the metabar coding, then the sample name should also go into experiment from metadata. And then other terms below here are mostly for sample metadata. And this is how you read these columns, and then those term names are divided or organized into separate sections. So first section is the project, so these are to describe about the project itself, and then the terms to describe about how you manage your data, sample information, how samples are related to each others, for example, control samples and batches of extractions and so on. Then information about sample collection, storage, and so on. So this will go down all the way to DNA extraction, PCR, bioinformatics, and so on. This column D here is the description of each term. Then we have requirement levels. There are 4 different levels. There's a mandatory, highly recommended, and optional. Some of the mandatory terms have conditions which you can find in this next column, requirement level conditions. This negative control type or positive control type are only mandatory if SAMP category, the term one above, SAMP category was negative control or positive control. Next column here is a term type, describing the types of terms. We have various types such as free text, controlled vocabulary, Boolean, numeric, and so on. When it's a numeric or integer term, then units are very important to make sure that all the data will be machine readable. So some of the terms we specify the units. For example, this one here is a sample storage temperature. We want it in degrees Celsius. But then some numeric variables, we allow users to select the units. For example, this one here, sample size. In here, under unit, you can see that we tell users to select from sample size unit, which is the next term here, unit of sample size, and then this is a control vocabulary. How to find the options of the control vocabularies are in this column K here, controlled vocabulary options. In this case, sample size unit, we provide various types of unit where users can select from. Apart from this type of unit terms with a controlled vocabulary, we also have multiple term names with a controlled vocabulary term type. For example, this one is an assay type where you can select either targeted or metabarcoding. And if it's something else, you can also specify using other option. These control vocabularies are very important in order to enhance the machine readability. The next column is example. Then you find the sample type specificity. This is about the sample types. So common eDNA sample types include water, sediment, air, soil, and so on. And some of the terms are applicable only for specific sample types, which is or indicated here. For example, this part here, they are all about filtration methods, so pump flow rate and filter pore size and so on. And those are applicable for air and water samples, but perhaps not for soil. And this is why we specify that this is for water and air samples. Next column here is the source of each term. There are existing standards such as Darwin Core for biodiversity data and MIXES for molecular data. These are well-established international data standards and extremely useful resources for us to use as a starting point to develop the FAIRe checklist. So we try to use them as much as possible. However, we also found that we cannot fully describe eDNA workflows with these existing standard terms. So we created new terms to fill the gaps. Under this column, this source column, you can find which terms are newly created by us and which terms are from Darwin Core, MIXES, and other resources. If we took from existing resources, then you can find the URI here. If we made any modifications in existing resources, then we specify here. For example, in this case, we modify description so that eDNA practitioners can easily understand, or it can be applicable for eDNA context. So I walked you through all the columns in this checklist now. So let's scroll down all the way to the bottom. You notice there are a lot of terms here, all the way to this row number 338. But many of them are actually not applicable for your workflow. For example, if your project uses a metabarcoding approach, then all the terms from the targeted assay section will be irrelevant for you. There are also a number of environmental variables, which you can see here in the bottom section of the checklist. And you might not have many of those data, and in that case, you don't need to include them in your metadata. You can also see that most of these numeric terms have unit terms. And so once you take these factors into account, the number of terms relevant for your project will be much lower. The template generator tools that we are going to use now will take your workflows and sample types into account and provide only the relevant terms for your custom templates.